Christiane started her career in Germany at the University of Tuebingen and the Freie Universität of Berlin, where she established right from the outset her research interests in assassin bugs. And a bit later on, after finishing her PhD, she moved to the American Museum of Natural History, where she turned her attention to mirid bugs. But since 2007, she's been looking at all of these heteropterans groups at the University of California, Riverside, where she's professor in the Heteropteran Systematics Lab. Her research focuses on using morphology and molecular methods to study the evolution of behaviors such as host choice and prey specialization. And I'm not going to say any more because I think Christiane is going to tell us far more detail in her talk, which is intriguingly titled From Hunters to Herbivores, Systematics and Evolution of Assassin and Plant Bugs. So over to you, Christiane, and thank you.
Wonderful. Good morning, everyone. Thank you very much for the very nice introduction. And of course, I am very honored to be speaking here today in front of all of you. So thank you very much for the invitation to beautiful Glasgow. So I want to start off speaking very briefly about this title slide because it combines two of my most favorite insects. On the left, you see an assassin bug that is known as a corpse camouflaging assassin bug because they pile ants, they suck dry onto their backs, which really helps them to be camouflaged and sneak up on more ants, but then also really be not visible to potential predators. And my title is, my talk is titled Hunters to Herbivores. So what you see on the right is actually a plant bug, a mirid, but I'm cheating a little bit because this particular plant bug is actually not a plant feeder. It does actually scavenge on carnivorous plants, which to me is a pretty amazing, outrageous behavior for a plant bug to engage in. So for the next 50 minutes or so, I want to take you to the world of Hemiptera and specifically Heteroptera, the true bugs, which are part of the fifth largest insect order, the Hemiptera, that also includes cicadas and a whole bunch of other lineages that are all strictly plant feeding. So it is really only the true bugs that diversified in terms of their diets and went wild and became predators and scavengers and fungivores and you name it, essentially. What attracted me to that group now a long time ago is there's lots of species, about 45,000 described species in that group. But we also know there's lots more undescribed species, and I've always been attracted by that unknown out there. And similar for our understanding of evolutionary relationships among these groups, we had some emerging ideas about how the big groups might actually be related to each other, but there were a lot of unresolved areas. And that is a shame because, as I mentioned earlier, true bugs are really fascinating. They include, for example, bed bugs, which have become a really bad reputation, obviously, due to their resurgence over the last couple of decades. But there's also a lot of really interesting evolutionary strategies in a group, such as paternal care, maternal care that evolved many times separately in different lineages. We have some assassin bugs that inhabit spider webs and they will actually feed on spiders. And we're going to be hearing a little bit more about that later on as well. And then we have a lot of really brightly aposematically colored species in true bugs. And we know that at least some of them are capable of sequestering toxins from the plants they feed on. And beyond just that pure interesting evolutionary stories in my mind, there's also actual relevance in terms of the economic importance of true bugs. So a good number of them are agricultural pests. They can be really quite damaging to a whole range of different pests around the world. I've already mentioned the nuisance pests, but there's also good guys, they're natural enemies, not maybe so much the assassin bugs I'm highlighting here, but there are certain groups of plant bugs, for example, that are actual credible biocontrol agents. And then, of course, we have the super bad ones, which are disease vectors. You may have already heard a talk about kissing bugs or Triatomine yesterday afternoon. And with all that in mind, it is really important to actually know what species are out there, to be able to actually identify them, but then also to have really good understanding of the evolutionary relationships across the entire group, which then can be translated into a meaningful classification. And beyond that, I'm really interested in using these emerging phylogenies then to overlay them with other sets of natural history data. That could be just distribution, but it could also be behaviors, including diets, for example, or other kind of traits. So just continuing my little introduction of true bugs, they're relatively complicated. There's a whole bunch of different what we call infraorders. Here you see the three small ones, as we call them, Anicos, Dipsos, and Geromorphs. The numbers you see in these square boxes are the numbers of families in each of these units. So not very many in those. Then we have the Nepomorpha, which include all the aquatic bugs, really. You may be familiar with water scorpions or giant water bugs. And then we have all the rest of the true bugs. And this is really where the greatest diversity is, not only in terms of the number of families. You see 17 here for Simicomorpha, 42 for Pentatomomorphs. Those are the stink bugs and related groups. But this is also where pretty much all the species diversity is across true bugs. So when I started working on a group, we had very rudimentary ideas of what the relationships might be between these big infraorders, these big groups. But over the last 15 years or so, we've really come to what I think is a pretty good consensus. We now have very stable analyses that give us pretty much the same results over and over again, which is what you want to see in phylogenetics. And this latest analysis that you see here includes actually a lot, in my mind, of the families, 74 out of the 88 recognized families. There's a bunch still missing, but those are often monotypic families that are only known from the holotype and may have only been collected once at all. So we're missing, obviously, a lot of those. But these trees now show us that Nepomorphs, the aquatic groups, are the sister group, the earliest diverging lineage in all of the true bugs. Then we have these little groups, Anicos, Dipsos, and Geros together. And then we have Leptos, the sister group, to all the really big, kind of most diverse groups here. This result isn't particularly new, I want to say, in the set of overall relationships. This is something we've already shown in 2019. At this point, it was based on Sanger data plus morphology, really, but we had the same set of relationships. And at that point, we use it to really trace the diet evolution of true bugs across the entire system. And we see here that the most common recent ancestor, like the last ancestor of all living Heteroptera true bugs, made the switch from being plant feeding, which is the condition in all the outgroups and all the more distantly related groups, to becoming a predator. And that this diet was retained along what we call the backbone of all of true bugs, including in the last common ancestor of the Simicomorphs that include plant bugs, mirids and related groups, as well as assassin bugs. From that, we can conclude what you see here in green. Those are the plant-feeding lineages that the plant feeding we see today in stink bugs, which is also a big group of pests, and in the mirids are independently derived evolutionary transitions. We also looked at microhabitat evolution at a very coarse level, just trying to figure out what along the backbone of true bugs really happened in terms of terrestrial, semi-aquatic, aquatic habitats. And we could show that the last common ancestor of true bugs was terrestrial, that was retained across the group. And then in the last common ancestor of Pentatomomorphs and Simicomorphs, again, these really speciose groups, we can show that plant association evolved as the dominant microhabitat type. So that also means that really the two groups I'm going to be talking about today, the Reduviidae, the assassin bugs, and the Miridae, the plant bugs, are part of this plant-associated group. So some just use the plants as sort of the background, essentially, where they hunt for the prey they're feeding on. but then others presumably evolved in much closer association with these plants, as we see in Miridae or plant bugs. Before I get into the nitty-gritty of assassin bugs and mirids, I want to step back briefly and say a few more words about who I am, where I come from, and why I'm here, essentially. So I grew up in a very picturesque little town in southwestern Germany called Schwäbisch Hall, which is sort of a little bit of a tongue twister. My parents are not entomologists. They don't have a university degree or a high school degree, so I'm the first to do all of that in my family. But they've been always extremely supportive of me doing what I wanted to do. And I knew I wanted to study nature and discover things and travel the world, essentially. And that's really what I've done over the last decades. And I now live in Southern California, very close to the Citrus Historic State Park, so surrounded by citrus trees, and mostly in very hot climates, which is sometimes fun, sometimes not so much. As was mentioned earlier, I did my undergrad at a university in Tuebingen, which was a very pretty old university. I was lucky enough to do a study abroad year at the University of Queensland in Australia, and this is where my interest in assassin bugs really started, because I was allowed to go into the university collection and start rearranging and identifying the assassin bugs, which was really cool. So that was a great experience and starting point. I then did my master's in southern Brazil, looking at the natural history of the assassin bugs I found in this area. Continued on to the Free University in Berlin, where I started working on assassin bugs for real. I want to say I started doing phylogenetics. And then continued on to a postdoc where I started getting into plant bugs at the American Museum of Natural History in New York City. And finally, starting in 2007, I became a professor at the University of California, Riverside. And this is where I'm leading what we grandiosely call the Heteroptera Systematics Lab at UCR. You see some of the field teams that have enjoyed collecting in Southern California over the past. And really, this is one of the sort of dominant themes in my lab. I want to say we're all very fieldwork focused because despite the value of natural history collections, of course, for morphological research, but then also the increased use of museum specimens for molecular approaches, there's still a lot of insects out there that have not actually even been collected. And I'm going to be showing that in a few cases throughout my talk. So to me, fieldwork is a really important part of species discovery and documentation. We're doing quite a bit of phylogenetics, but then, as I mentioned, we're really interested into these beyond aspects as well, where we're using our trees to really use them to better understand the groups we work on. Here are some of my mentors. So I want to say I didn't have a typical entomological background in a sense that my friends and closest mentors were fish people. My major professor was a nematologist at a Free University in Berlin, but I was also surrounded by some Heteropteras, and then Toby Shu became my postdoc mentor at the American Museum of Natural History, and that was really instrumental. When you look at all these faces on the screen, it is clear that they're all fairly white male dominated, of course, as you would have expected from that time. So I am very happy that my current lab looks a lot more diverse. And this is something I feel very passionate about, trying to grow as much as I can. In terms of teaching, what I do, I teach insect biodiversity, as you would expect from a systematist, insect evolution, a separate class, systematic theory to some extent, and an entomology capstone for our undergrad majors in entomology. But a few years ago, I also created and now am co-teaching a science communication and insect outreach class that I feel very passionate about. And also, just as a heads up, we're offering a TrueBug short course that's not part of the university, it's part of a current grant. It will be next offered in June 2026. So if you or your students are interested in learning more about TrueBugs, keep your eyes open for postings. This may be the last time this TrueBug course gets offered for now. And you get to hang out in these beautiful environments, do light trapping at night, and find magnificent assassin bugs and more. Okay, I mentioned outreach a little bit. So this is something I got into right after we sort of came back after the pandemic. I was essentially saddled to really figure out our outreach program going forward that had pretty much died over the pandemic. So I tried to really invigorate creating new displays that are being displayed all throughout our department. I was liaising with our Natural History Museum Club, an undergrad student club, to really, come up with better activities and displays as well. We created the outreach class as part of these efforts. And then I rebuilt our what we call insect zoo from containing 3 species, this is what we had right after the pandemic, to now a little bit more than 30 different species of insects we take out to outreach events. Okay, with that, to assassin bugs. This poster is a poster showing a lot of different subfamilies of assassin bugs that a former undergrad of mine put together. And this is a large group. We're talking about 6,800 different species, a little bit more than 20 subfamilies total, which is a pretty large number for any family. Of course, they include the kissing bugs, which is the really most economically important of all the different groups in assassin bugs. They're very distinctive morphology in different lineages. And above all, there's a lot of really cool natural history or biology. And this is really what's attracted me to this group. So there's aggressive mimicry that is found in Nigeria, and John Harrity was the witness. I cried when I saw that for the first time, because there's only some historical notes on that really. And seeing a behavior like that for the first time out in the field, that is just entirely incredible. So we saw these Heteroptera assassin bugs feeding on cotton stainers. There's a phenomenon called sticky trap predation, where assassin bugs will collect resins on trees, and then that really allows them to hold on to their prey organisms much more easily than they would be able to do otherwise. I've already mentioned corpse camouflage before, another crying moment for me out in the field to see that for the first time. That was really incredible. And then also maybe a little bit more simple, just ambush predation with amazingly camouflaged insects really that blend into the background in a way that is very difficult to see them. So very fascinating group. And I kind of make it sound like we actually know a lot about the natural history of different species. And of course, you can imagine we do not. So the about 150 species of kissing bugs are relatively well studied, but then all the rest of the assassin bugs, what we know are just really little glimpses of what actually might be out there. So for me, working on assassin bugs has now been a 25-year journey, which is kind of a little intimidating to say that way. What you see in the lower left here was the title of my defense talk, dissertation defense talk in 2003, of course, in German. And what I had worked on during my dissertation was really coming up with a morphology-based phylogeny of assassin bugs. Our molecular data show me I was right on certain things. I was very wrong on others, but I think that is to be expected. Let's put it that way, I'm proud of the things I was right on. And one of those is that Reduviidae, the nominal subfamily, is not what we call a monophyletic group, which is a natural group that evolved once and all of the descendants are included in that group. But instead, they're, as you see with the red arrows, they're scattered across all of the rest of the assassin bugs. And this is something that's become more and more clear with us including additional taxa, additional species, and using different data sets, such as molecular data, simple Sanger loci at this point, no morphology. There's even a lot more of these red arrows that show us that Reduviidae are clearly a problem, and someone will have to do something about that problem at some point in the future. And that was in 2012, so we said someone will have to do something about it. And it only took us about 13 years to actually do all of that. So our problem was that we wanted a really robust phylogeny, very data rich. So we wanted genome level data essentially, or at least subsampled genomes, so 2,200 loci roughly in this case. And it wasn't really enough to just come up with this big molecular phylogeny, but we also needed to include morphology. And this took an extra year and a half, I want to say, because in the end, what we had to do is fill out 21,000 little cells in a big morphological matrix that then was analyzed together with the molecular data to come up with this publication that was published in Systematic Entomology last year. And we're very proud of it, not a question. So what does that tree tell us? Well, first, and this is a bit of a downer, the tree isn't really all that different from the tree we saw in 2012 in terms of overall relationships. We do have much better representation of all the different lineages of assassin bugs, so that's good. And we also have much better support values across the entire tree, so that's great as well. And in the end, we took this tree, translated it into a classification. So we now officially have 19 subfamilies and 40 tribes of assassin bugs. We described 3 new subfamilies, and really rummaged around all the rest of the assassin bugs such that we now actually have a monophyletic subfamily, Reduviidae, by really combining a whole bunch of subfamilies together and describing others as separate entities. So how long it will take for this classification to get universally accepted, we have no idea. It will be a while, but we're working on a publication, putting together an identification key to those, and we hope that really will help for people to better recognize these groups. Okay, to finish off assassin bugs, I just want to give you 3 little, I call them vignettes, on cool behaviors really in assassin bug world. And we're going to be starting off with kissing bugs, where a few years ago, I was collaborating on the first UCE, ultra-conserved element phylogeny, actually of true bugs, I want to say, in general terms, focusing on kissing bugs. And what we did in that analysis is really combine large molecular data sets with Sanger data for a lot of the species we couldn't really get new material of. And we showed that kissing bugs really are in trouble, a little bit similar to the way the overall Reduviidae are as well, in terms of one of the tribes being polyphyletic, again, no monophyletic groups, same as for all the big genera. So there's a lot of effort in the future that probably will need to go into, aligning these classifications with phylogenies. But in my mind, one of the cooler outcomes of this analysis was that we could show that this bug here, which looks a lot like an kissing bug, but really is a predator. They are found in nests of birds. They are the closest relatives to kissing bugs, which gives you a pretty good sort of transition from free-living assassin bugs, living in nests, but still feeding on insects and nests, into the blood-feeding behaviors we see today in Triotomini. The other cool thing about that tree in my mind was we could figure out the biogeography of kissing bugs much better than we've ever been able to do before. So the great majority of kissing bugs live in South Central and North America, but there's literally a handful or so of species that are found in the Old World, anywhere between the sub-Indian continent and Northern Australia. So for a long time, I was curious to see how did that happen, and was it one transition from the New World into these Southeast Asian areas, or did it happen multiple times? And our tree is really clear that the Old World was colonized only once. And to make it actually even more exciting in a way is there's one species that is worldwide. It's also found in the New World. And we now can show that that is actually a secondary invasion of the New World from the Old World. Okay, with that to a completely different group and the evolution of sticky traps. Again, this is something I've been really excited about since my dissertation work, where this is my defense talk in 2003. I came up with now pretty disproved, I want to say, ideas on how these sticky trap behaviors may have evolved in Heteroptera. But I've remained curious, obviously, about better investigating these evolutionary transitions. And we did that based on a phylogeny that was published quite a while ago, but that allows us to see that these different behaviors of using sticky plants, sticky substances, really evolved multiple times across that entire subfamily Heteroptera. So resin collecting and applying resin on your forelegs evolved 3 times independently. Having your own glands that produce sticky secretions, that only evolved once. And then being associated with plants that are fairly sticky, as you see down here in this little video. So there's a Celeopus, a assassin bug, just walking around on these sticky plants. And then it will scavenge on insects that get stuck on these plants, as well as it will use the resins to make itself a little bit more sticky along the forelegs. And we can show that this behavior evolved a minimum of 4 times. There's some out there that's just been discovered, so that number is just going to go up. Why that is, why there are so many times of these associations, we do not know, except the entire subfamily is very closely associated with plants, typically. And then finally, but not last, I want to say, speaking about stealthy predators, the subfamily Amazoni. So if you remember the overall number of assassin bugs, it was close to 7,000. So 1 in 7 assassin bugs really belongs into this Amazine group that are called the thread-legged assassin bugs. So this is an immensely successful subfamily. We sadly don't really know that much about behaviors, again, of the majority of species, no economic importance, no one really studies them. So what we decided to do is try to use morphology to really infer natural history and behavior for species in a group for which we don't have data. So what we did in this case is we looked at pretarsal structures that are involved in web-associated behaviors. So what you see down here is one of these little Amazines hanging out in a spider web, having no problem walking around in a spider web. And then in this case, eating the spider, which is not very nice, but that's what a lot of them do. Some of them only seem to be stealing the spider's prey and not go after the spiders themselves. And we found that these pretarsal structures that seem to be predictors of if an assassin bug can live on a web or not are really quite widespread within the subfamily Amazoni. So we think these web-associated behaviors are likely very widespread. They're not particularly easy to observe just because they typically happen in tropical rainforests, somewhere around the bases of trees in darkness, pretty much. So not a lot of actual natural history data, not as much as we would like to see. Okay, so what's next in assassin bug world? Well, we're in the process of describing a whole bunch of cool looking fossils. We're revisiting diet evolution because there's a lot more insights we can gain, we think. And then I started co-advising a PhD student from Oxford Brookes, who's looking in what we call FLABs, which stands for feather-legged assassin bugs, the Heteroptera, and he's been doing some really cool work in Australia. So stay tuned. Hopefully he will come to the next meeting next year and tell you all about that at that point. Okay, with that, I want to switch over talking about plant bugs for a little bit. So now we're talking the presumably herbivore side of things. So Miridae, they're a megadiverse insect family. There's lots and lots and lots of species, many yet to be described. The classification has been relatively stable, I want to say, mostly because no one's actually worked on it. They are of economic importance. So there's pests, there's beneficial insects in a group. There's a very active taxonomic community out there. So there's lots of people actually discovering species and describing them, which may sound a little weird, right? Because plant bugs around here, they don't look all that attractive. But you really want to go into the tropics, and this is where some of the really wowing species are found. And as you would expect, there isn't really actually enough phylogenetic information. Out there to really make meaningful inferences on the evolution of the group. And what in particular has bugged me for the last 20 years or so is that there's a wealth of spatial and host plant data for Miridae, but we're lacking the phylogenies to really do something with it. So we decided we need to actually change that. So the reason there's so much spatial and host plant data for Miridae are two large National Science Foundation, US National Science Foundation projects. One looking at two of the largest subfamilies. That is the project I was a postdoc on. And then we had a follow-up project where we were really trying to database as many of the plant bugs and other groups in North America specifically. So we now have close to 500,000 of specimen records databased, which is a pretty good starting point. We have geographic and host plant information for more than 10,000 species. And as you would expect, given where the funding sources were, we have the best coverage in the Nearctic. So we've shown before that areas of endemism for Miridae across North America are interesting in the sense that they're relatively small, and there's actually a lot going on in the Western Nearctic, specifically in California, or the so-called California floristic province, that we call a plant bug diversity hotspot. But we do not actually know anything about where these different species came from. Did they originate in the California floristic province, or did they actually colonize that area? And similarly, when you think of plant bugs, you think plant feeding. Well, that's true for a lot of them that are phytophagous, but there's also lineages that are strictly zoophagous, meaning they only feed on other insects. And then there's a whole bunch of species that seem to be doing a bit of both, depending on what condition maybe the plant is on or how many other insects to feed on may actually be around. So we're very interested in trying to figure out these patterns of host plant evolution, but then also diet across the entire group. When it comes to phylogenetic hypothesis, well, there wasn't really that much done, really. And the problem with these phylogenies is they don't really agree on even something like the earliest diverging lineage of plant bugs. We don't really know. Some of the subfamilies seem to be well supported as a monophyletic group, others are not. But again, we don't really actually know. And then these uncertainties really mean we can't use these trees for anything in terms of downstream analyses, figuring out divergence dating, those kind of applications. So we're lucky enough to get more National Science Foundation funding a couple of years ago. The grandiose title of this project is Spatial Phylogenomics and Diet Evolution of the Megadiverse Plant Bugs. I'm leading that together with two co-PIs, one postdoc, 3 PhD students, a master's student, and a whole bunch of undergrad students are part of this project. And as you would expect for a project that is meant to be really worldwide, we do have a lot of collaborators on a project as well that help with various aspects from fieldwork logistics, providing specimens to really helping with identification and interpretation of the trees that we're generating. So the project has 3 overall goals. We're trying to put together a really robust, well-sampled phylogeny using ultra-conserved element loci. I will give you a little bit more detail on that. So in the last couple of years, we've been going through our freezers and we've been going through natural history museums to pull all the different species that we think are important. It's a lot of work. We have 8,000 what we call batches of specimens we've collected in the lab. And just going through those and trying to identify them in a group that's as diverse as mirid, it is quite daunting. So we really have done a lot of that work ourselves, but also have gotten a lot of help from collaborators. We have specimens in the pipeline for more than 600 genera, about 50% of the genera of the 2 largest subfamilies. And we've established protocols that allow us to not only use ethanol preserved specimens, which is much easier to work with, but then also dry specimens as well. We have more than 1,000 taxa sequenced at this point and 800 included in analyses. And you're going to be seeing some of these emerging phylogenies in a little bit. The second goal of the project is really looking specifically into the Western Nearctic, because as I mentioned, we're really interested in figuring out where does that diversity in California come from. So we decided to focus on specific genera and do deep dives into those and then try to figure out both host transition patterns, but as well as historical biogeography. This is a little progress chart on the side where we're going with the different genera, and there's no real results for that part yet that I'm going to be talking about today. Similar for Project Goal C, this is where we're going to be doing phylogenetic diversity studies on the California floristic province specifically, because if you look at a map of ecoregions of California, this is a very complicated area. So there are so many different ecoregions, meaning there's a lot of different plants, and that translates to a lot of different plant books. And we've known that for a long time, that really California floristic province is a hotspot. So there are about 1,000 described species recorded from California. More than 30% of them are strictly endemic to the California floristic province. We have a lot of really good data-based records. And what we decided to do for this part of the project is go after freshly collected ethanol preserved material, because the methods we're going to be using to do the analyses are very sensitive to branch length estimation. So in my mind, this is a really good excuse to do new field work. And this is exactly what we've been doing for the last 1.5 years. We did about one trip a week, some of them only one day, some of them multiple days, starting in early March in Southern California and essentially working our way up north and up into the higher elevation. And yes, there is snow in California. In fact, there's more snow I've seen in California than in Germany, where I grew up. You have to go high elevation, but there's lots of snow. So those are some of the areas we've collected in last year, and then we added a lot more areas in 2025, as well as our northern team based in Sacramento added all these blue areas as well. Photos of the field teams, very important. So we involve undergrads a lot in our field work. The grad students are leading some of the trips at this point. And it's been just amazing to travel to all these places in California that, well, I've never been to before. So it was a great opportunity for me as well. The way we're collecting plant bugs is we use a beating net and a stick, what we call a Herbivore aspirator, just to suck up insects into a little glass tube, keep our finger on it, and then spit them into the collection vials, really. We typically keep ethanol and ethyl acetate vials, just because you put a mirid in ethanol, it will lose all the appendages, and they can be extremely important for diagnostic features. So we try to keep some dry so we can actually identify them, which is important. We're keeping good records of the host plants. We're not actually collecting hosts anymore, just because websites like PlantNet, for example, have become so good in identifying all the commonly encountered species in California, and that's typically where our plant bugs live, that we don't actually really need to collect plants anymore. And over the last 1.5 years, we've collected a lot of localities, a lot of different field hosts, and we've done a lot of identifications on these critters. Here are some of the highlights, some of my favorites. Species of the genus Macrotylus. They sit on very nice smelling plants like sages over here, and they're just very cute little bugs. And then some of them are just stunningly colored. So this takes away with that all mirids are kind of boring in a way. And this may be one of my all-time favorites. We actually know exactly where in the phylogeny it goes to, which is cool. It's related to a whole bunch of much bigger plant bugs, none of which have these outrageous antennae. But there's just one bug where the males in particular decided to inflate their antennae and shrink their body size, and is feeding on these very hostile looking plants that are found in the deserts of Southern California. Not very appetizing looking to me, but a really adorable bug. We also were fortunate enough to do a really cool trip to South Africa, which is another hotspot for mirids. So a lot of mirid hotspots are in the Mediterranean type regions around the world. South Africa is one of them, and probably the least well studied overall. So we took that as an excuse to really help us with kind of better understanding of the different lineages that occur there. We had a really good field season or field trip, found some relatively large plant bugs, a whole bunch of them, different species. And it turns out that genus doesn't even have a name. And you go, how is that possible? They are really quite obvious, but now they make a fantastic thesis project for one of And similarly, we collected what we call a mystery genus that we only, based on the phylogenies that came out a few weeks ago, now can confidently place into one part of Bryochroini. That looks very outrageous and is very unusual. It's a group of bugs that typically live on sticky plants, but this particular species is found on Kalanchoe of all places, so of all plants, which is not very sticky at all, really. Very strange succulent. And last but not least, we were able to collect both described species of a genus that's called Miridae, that is known to only live on insectivorous plants, Riverside, which when you look at the stickiness of these plants, this is quite amazing for those little bugs to walk around on those. Actually, there have been studies done on how they do that. So they lift their bodies up a little bit and essentially just make sure they're not really interacting with the sticky secretions. But there's a similar behavior that occurs in species in Australia. And because we have both material of the South Africans and the Australians, we're now able to figure out, is that one evolutionary origin of moving onto carnivorous plants, or is that something that evolved multiple times? So our protocols, I'm not going to be going into any detail in, but those were the first of our emerging trees, fall 2024. Not all that impressive yet, 107 in group, meaning mirid species. But what is really striking is overall support, obviously, in these trees is really quite well. We're not quite, we're in the process of analyzing our data all sorts of different ways. So there's a lot more analyses that need to be done to really trust all the results we're getting in these trees. But overall, things are looking really quite good in a way. And the trees have been growing ever since to 1615 taxa in spring 2025. And we're now at 806 taxa. But we also felt earlier this year that the trees now are actually starting to be good enough for us to look into diet evolution across all of Miridae at a relatively coarse level for now. So just to remind you, phytophagus, zoophagous, but then there's also microphagous groups feeding only on fungi. And then we have lineages that are feeding on both based on availability in many cases. And here the somewhat surprising, I want to say outcome is the last common ancestor of Miridae was phytophagus. That part is not so surprising because this is what related groups are doing as well. Phytophagy was retained along the backbone of the group, but then zoophytophagy evolved 30 times across Miridae and zoophagy an additional 18 times, which is really quite surprising, I want to say. So in my mind, that probably makes it a family with the most diet transitions really out there amongst insects. So this could be a very interesting system to really further dig into what may have driven that. A very brief thing on Bryochroini, which is one of the most economically important subfamilies of Miridae. They're the good, they're zoophytophagous, they're used in biological control. And then there's some serious plant pests out there, Helopeltis, the Aurora being one of them. So one of the more shocking things is Bryochroini that we often refer to as a group. Well, they're not monophyletic. And even Dipsocoromorpha, the biocontrol agents, really are not. So they belong to the two earliest diverging lineages of all of mirids, which is intriguing. And as I mentioned earlier, because we have samples of the Australian as well as the South African carnivorous plant feeders, we can now show in this little inset down here, those two lineages in blue, they evolved independently in Australia and in South Africa. Last but not least, Miridae, the most economically important group of plant bugs. They're going after a whole range of different crops, including California, for example, strawberries is a really big one. And what is intriguing too about this group is there are some genera that are extremely speciose. There's 700 species of Phytochorus, and some of the other ones are really big as well. So one of our ongoing things here is to try to figure out really what has spurred the diversification of these different lineages. The Miridae phylogeny, as it stands right now, based on our data, again, is a mess, as you would expect. The phylogeny does not translate to the classification. And in particular, when it comes to Herdoniinae, for example, the end-mimicking Miridae, you see that they're scattered all across the system of Miridae. So people just put them together into one group, into one tribe, because they look like ants, but they really are not closely related to each other. So I'm finishing off with this last slide, Miridae and plant cell wall degrading enzymes. And I'm not going to be talking about PG tips per se. I am talking about polygalacturonase, PG family of genes, where we know, based on a publication that came out a number of years ago, that there was a horizontal gene transfer event from fungi to certain beetles, but also to Miridae. Or at least this is what that study claimed, even though they really only looked at 4 different species of mirids and no outgroups. So now, with the trees, we've been developing the whole genomes, low coverage whole genomes we've been developing. We can actually dig into that much more closely. So as of yesterday, I can confirm that the horizontal gene transfer actually happened in the last common ancestor of Miridae, because their close relatives, Tomastocorids, Tingids, don't have them. And we can also see the first glimpses of PG family expansions, duplication events really in the genes, in those groups that are phytophagous. It's very early stages, but what we think we're seeing is that Miridae, this really super economically important group, where species can often feed on a whole range of different host plants, they seem to be having very, very high numbers of these PGs, which in part may explain how they've become so successful. So the next steps for us here are looking into, was it one or multiple HTG events that happen in mirids, and then trace the individual evolution of individual PGs to really better understand the dynamics of these genes across the entire group. And with that, I would like to thank all of you for being patient with me while I talk about true bugs, and for your attention.
Well, thank you very much. That was a fantastic talk. Just what a broad breadth of things you've studied, and I didn't know very much at all about those groups, so I feel much better informed there. Can we open up for questions? Does anyone have questions?
Thanks very much for that. You mentioned in the talk that in your thesis you had various hypotheses about the evolution of the sticky ones that that you subsequently disproved. What were they, and how did you disprove them?
Well, so at this point, I had a very simple phylogeny, right? It didn't include a lot of species of that particular subfamily, Heteroptera. So I included some of the resin bugs, for example, but I wasn't able to include all of them. And based on the morphological analyses, it seemed to be a very nice transition from you collect resin first, you then transition to bugs, behaviors of normal plants that have sticky trichomes associations, to then the most highly derived group, it seemed, were the ones that have sticky glands themselves. Well, with adding a lot more taxa and a lot more data, in particular molecular data, it turned out, yes, the resin bugs are somewhere around the base of the tree, but it's not as linear as a transition as I supposed at that point. So it was just better taxon sampling, really, and a different set of phylogenies, really, that disproved my dissertation hypothesis. Gone.
Hi, Christiane. Thank you for talking to us about Hemiptera. I wanted to know, and forgive my ignorance about bugs, Are there any correlations that you see between social behavior and diet in any of these insects?
That's a great question. So, okay, social behavior is not something that's been studied very much. I want to say across bugs in general, and I'm a little cautious here because it hasn't been in mirid, certainly, so I can't say anything about that. When it comes to assassin bugs, there aren't very many what you would consider social, subsocial, whatever species. They're really only found amongst the millipede assassins. So that's a group of assassin bugs, 600 species, all specialized to feed on millipedes. And taking down a millipede is, not an easy thing. So it seems like there is more maternal care in that group than we typically see in assassin bugs. And it seems like the babies often stick together for long periods of time. And then they seem to rely on females to help take down a pretty substantially big millipede. And then they would have communal feeding on this millipede. But that's really the only thing I can really think of that would be truly, not even truly, but somewhat social, I think.
Hello. First of all, fantastic talk. Thank you. It was a great way to start the Wednesday. This is a bit of a fun question for you. Out of all the assassin bugs, do you have a favorite group or species and why?
So I want to say, yeah, when it comes to assassin bugs, I think I mentioned that multiple of them have me made cry before, right? But I think really that corpse assassin, it is one of the coolest stories, really. And that's where you wish you lived in Singapore or somewhere in a tropical country where you can actually explore them on your doorstep every day, which sadly for us, I mean, our tropical trips have always been a whirlwind of trying to do too many things, like, collecting as many bugs as you can and then doing some little observations here and there. But a former grad student of mine is a curator at collection at NUS, National University in Singapore. And he now has the ability to actually go right outside of his door and really observe some of these critters. But yeah, they're pretty high up there.
Hi, thanks for a really interesting talk. What proportion of true bugs do you think remain to be unexplored in terms of alpha taxonomy?
Yeah, that's a great question. Obviously a difficult one. So I think in some of the large groups, assassin bugs, yeah, there will be a few, right? Few undescribed species, but there's also still a lot of synonyms. So species that have been described more than once because no one's actually really looked at them. So my predictions for a group like assassin bugs is it may go up another 10%, maybe 20%, but that's pretty much it. When it comes to plant bugs, and they are not the least studied group, I want to say, it's probably 50% at least are undescribed, probably. South Africa was a good example. There's so many of the specimens we collected, there's no way we can put a name on those just because they've never been collected before. There's no names for those. And then you go into the truly, the dark taxa, I want to say like Dipsocoromorpha, the minute litter bugs, highly diverse in the tropics. Right now, it's only about 400 or 500 species described. We describe probably half of them in my lab. And if we continue with the samples we even have just consolidated into our lab, we can easily probably go up to more than 1,000, 2,000. So yeah, those are just sort of some of the extremes, I want to say. Difficult.
Hi, yeah, I've got questions about how they attack, for example, the venom/toxins. Is there like any studies being done on looking at like how they differ or like evolve differently?
Okay, you're putting your finger into a wound because yes, a number of years ago, we were supposed to be really looking into that because we do now have, transcriptomes, genomes, some of them actually relatively decent coverage that we would be able to look into that a little bit more closely, but we really haven't done that yet. So there are studies that look into transition between assassin bugs and kissing bugs, going into the blood feeding avenue, for example, and all the transitions that happen with throwing out a bunch of genes and then inventing new ones, I guess. But when it comes to assassin bugs, we can see there are patterns. So some of the highly specialized assassin bugs, like millipede feeders, for example, they do seem to be having a proliferation of certain genes. Sorry, I can't remember the names because it was all very preliminary. Yeah, but then we didn't really pursue that at this point.
Sounds like a project waiting to happen.
Yep. Anyone?
I was wondering if you're planning on extending your sampling into other Mediterranean regions, like the Chilean one?
Well, I would love to. I do not know how easy it would be to navigate in terms of, collecting permits obviously have become a real contentious topic around the world as well. But yeah, I want to say Chile, as far as I can tell, there's been some plant bug work done in Chile, but not very much. So that certainly is a real big hotspot that needs further exploring as well.
Well, it sounds like we've got some new potential collaborations. Thank you again for a wonderful talk and for kicking off our Wednesday so well. Just to, before we go to a tea break, join me in thanking Christiane again.